CBSE NCERT Solutions by Vedantu. I am Ms. Gopika, your own biology master teacher. So today, as I told you yesterday in last class, I had informed all of you that today we are going to do NCERT line by line. Now for those students who do not know this, it is a very simple process where we go line by line of NCERT and I will help you understand the chapter by reading the NCERT. Now this way, what is the benefit is that you will be able to understand NCERT. Second thing is that you will never have the hassle of reading NCERT after the class because you read it with me and you understood each line of NCERT. So I'm going to just check if we are live yet so that we can directly jump into the class. Till that, give me 2 minutes. Okay, and those who are joining new to the channel, please do subscribe to the channel. Consider subscribing because this channel is going to give a lot of chemistry, biology and physics content. Okay, yes, awesome, we are live as well, right? Okay, let's go students. So we will go paragraph wise. Okay, I've already given the paragraph, so we will be going paragraph wise. Let's start the class. 1 second. Yes, okay, first paragraph. I'm just checking simultaneously if there is any issue there, okay, because the screen or anything like that. So I'll be simultaneously checking that. Yes, okay, let's start. So, what is exactly NCERT telling? Since the dawn of civilization, there has been many attempts to classify living organisms. Yes or no? From the start of civilization. So, dawn of civilization means from the start of civilization, there has been lot of research. Okay, lot of research to classify living organism. Because just imagine there are so many living organisms and none of them are going to get classified. Everyone is calling whatever names you want, like you look at the dog and you're like Jimmy and then I call it Tommy. So that kind of a confusion, just imagine, no classifications are there, no phylums are there, no kingdoms are there, nothing is there, just randomly you start naming whatever you want to name. That will not work out. Yes or no? So from the start of civilization, people have been trying their level best ki, okay, what do I do with it? Do I put this organisms based on something? Do I put this organism based on what? So like that confusions kept happening. Okay, and there has been many attempts to classify living organism. It was done instinctively, not using the criteria that was scientific but born out of the need. Okay, the need. I have to keep changing the colors, so you will have to excuse me because when I underline here you will not be able to see. I think I'll make the pen a little thick. Okay, yes. So from the need, that is, students, imagine I have a need that, okay, cotton, okay, cotton plant is going to give me the cotton that is required for my clothes, for my other things. Okay, and then there is another person, let's say Tommy is there, and he is telling that, wait a minute, cotton actually I can use it for something else. So depending on each of our needs, we started classifying organisms. So the first ever earliest classification, right, was based on food, shelter, clothing. Okay, food, shelter, clothing. I hope the screen is visible, right? I'm just checking how it is viewed for you people as well. Yes, it is perfectly clear, right? Perfectly clear. So depending on ki, okay, this is going to give me food, I will categorize them into food. This is going to give me shelter, I'll put them into shelter. This is going to give me clothing, I'll put them under that. But just think about it. As a science student, would you agree for a classification like that, telling that, okay, this food, see, some people actually consume some kind of food but we do not consume that, right? Now because of that, I do not want to classify them under food. Now what to do, ma'am? Okay, now what to do, ma'am, right? So depending on non-scientific classification, the first ever classification was based on food, shelter and clothing. Which would you agree as a science student? Yes or no in the chats? Yes or no? Yes, ma'am. Sukanya is telling, no, ma'am, I will not agree. How can they classify it into food, shelter, and clothing? I will not agree. Okay. Then the first ever scientific classification that came, okay, first ever scientific classification that came was the classification by Aristotle. So always remember, the question will come, which was the first scientific classification? Your answer should not be food, shelter, clothing. Okay, that is not a scientific classification. That was like, oh, depending on my greed and need, I will just classify the organism. The first ever scientific classification was done by Aristotle to attempt, like he wanted to be like, okay, wait, let me give it a try. Let me try to put a scientific classification. And what did he do? He used simple morphological characters to classify plants. That is, he looked at the plant is short, so he's like, wait, you're a herb. Okay, then he noticed that the plants are in a medium size, so I will call it bush or I will call it shrub. And then he noticed that there were tall plants, so I will call it trees. Very simple classification, yes or no? So the classification was based on simple morphology. So the first ever classification brought about was by Aristotle. Okay, Aristotle brought about the first scientific classification. Clear? Okay, first scientific classification. Now, how did he classify? He did the classification based on, he took plants. Okay, he took plants and he divided them into three categories: herbs, shrubs, and trees. Herbs, shrubs, and trees. Over herbs, shrubs, and trees, he did the classification. Then obviously we have animals. How can we forget the fact that we have animals? I'm so sorry, students. Okay, how can we forget the fact that we have animals? So he classified animals based on what? Based on if they have RBC or if they don't have RBC. A simple classification, right? Like, do you have red blood cells or do you not have red blood cells? Depending on that, he did the second classification. Okay, so second classification was animals based on, students, if they had RBC, it was called as enaima. Okay, and if they do not have classification, that is, sorry, if they do not have RBC, it was called as enaima. Absent A, enaima. Done. Okay, so animals were categorized into two categories depending on if they have RBC, if they don't have RBC. So if they had RBC, it was called as enaima, EE, and if they did not have RBC, it was called as enaima. Sorted? No confusion here, no? Don't write RBC, I mean RBC you write, don't write RCB. Okay, this was the first ever paragraph. Okay, this was the first ever paragraph. Clear? Now let's move to the next one. I think I'll use the highlighter, so it is better than this underlining. Okay, I'll use the highlighter here. Okay, screen is fine, no? Yes, enaima and enema. Very good, Sukanya. So here we will use highlighter, students. Now, okay, yes. So here next classification came. Now after Aristotle, see, always remember, after one scientist there will be a chain of scientists, there will be a chain of zoologists, there will be a chain of botanists who's ready telling that wait, he has found something scientific, even I can find something scientific. And the next kingdom was the two kingdom classification. Okay, oh, I think this will not happen. Wait, yes, two kingdom classification system. Okay, this was the second classification called as the two kingdom classification system. Okay, lots of ads are playing. Yeah. Two kingdom classification system was simple. Again, it was Plantae and Animalia. There it was plant and animals. Here he just added the AE, Plantae and Animalia. Now, what was so different about this classification that we call it two kingdom classification? Because if you technically see, even Aristotle did two kingdom classification. He put plants, he put animals. But this classification came into the entire set of classification because of the findings or because of how it was little different. Clear? Okay, how it was little different. Got it? Yes, okay. So it was called as Plantae, it was called as Animalia, and was developed that include plants and animals respectively. Now this system, okay, this system did not, very important, this system did not distinguish between what and what? Eukaryotes, prokaryotes. How can I do, like, I want to make a class of boys and girls, and the first ever classification that I have to take is the gender, right? I have to categorize all the girls, I have to categorize all the boys. Okay. Yes, dear. Yes, Sukanya. I'm doing line by line of NCERT. Okay. Line by line of NCERT. Got it? Yes. Okay. 1 second. I think I have to get. Okay. Yes. Yes, Sukanya. It is line by line. Okay. So students, first ever classification that I have to do is what? I have to categorize boys and girls. And I did not do the basic classification of classifying them depending on their gender. So eukaryote and prokaryote means what? That is the primitive classification, right? Right? Prokaryotes means very primitive. Eukaryotes means a little more complex organism depending on if they have the nuclear membrane or not. So that only he did not do. Then he did not do unicellular and multicellular. How can he not do that? Like, we have to categorize. No, when I'm categorizing, I should be confident enough to tell all the unicellular is here, all the multicellular is here. Okay, my voice is clear, I hope. Okay, my voice is clear, I hope. Thank you. First ever classification that I have to. Yeah, voice is clear, no? Okay, so he did not do that classification. Unicellular and multicellular, he did not do. Okay, and eukaryotes and prokaryotes he did not do. So that was a problem. Next problem was he did not categorize photosynthetic, green algae and non-photosynthetic, okay, fungi. He put everything together. Khichdi he made, okay, that is photosynthetic, non-photosynthetic, everything was together. So here students, the drawbacks are very, very important. They are going to ask you the drawbacks. So if they ask you what is the drawbacks, okay, drawbacks of Carl Linnaeus, okay, Linnaeus, okay, here it is there. Drawbacks of Carl Linnaeus. Carl Linnaeus is his full name. Main three drawbacks was that first thing, he did not classify pro and u. Okay, he did not classify pro and u. Second thing, what did he do? He did not classify unicellular and multicellular. I'm writing short forms. He did not classify photosynthetic and non-photosynthetic. Now, how can you do a classification like this? But still, he actually made a lot of modification from Aristotle's time. So that difference is definitely there. Okay, that consideration is definitely there. Look here. Classification of organisms into plants and animals was easily done, was easy to understand, but a large number of organisms did not fall into either of the category. Yes or no? When you do such a broad spectrum of classification, right? I do such a broad spectrum of classification, like um, let's say some very like white and black. Okay, white and black are two basics color. So I'm categorizing my clothes into white and black. Now, those are a very big spectrum. White is there, black is there. Now, what about the browns? What about the pinks? What about the reds? What about the beige? They will not belong anywhere. So, like that same thing, when they categorized plants and animals, it was such a broad spectrum that so many things did not even fall into it, right? So, that was one problem, okay? A large number of organisms did not fall into the neither category. Hence, the two kingdom classification used for a long time was found inadequate, okay? Besides gross morphology, so he also took morphology, a need was also felt for including other characteristics. Like just looking at it, like, okay, this is this, okay, like this looks like this, so I will put them here. Not just looking at the gross morphology, they wanted the scientists to look at what and all. They wanted the scientists to look at the cell structure, okay? They wanted the scientists to look at the cell structure. They wanted the scientists to look at the nature of the wall, okay, nature of wall. They wanted the scientists to look at the mode of nutrition, like are they heterotrophs, are they autotrophs, what are they doing, right? Nutrition, habitat, much more, okay? Methods of reproduction, methods of reproduction, like that. They wanted them to not just look at the gross morphology, okay? Gross morphology wasn't the thing that everyone was looking at that, but that was just one layer of classification, okay? One layer of classification. They wanted the say the zoologist and botanist to go into the depth of finding the cell structure, mode of nutrition, reproduction and other things, okay, little more. Because sometimes what happens is when you're morphologically categorizing something, okay, when you're morphologically categorizing something, you will notice that anatomically they might be similar, but morphologically they might be different, okay? Morphologically they might be different, but anatomically they are a lot similar. So that kind of things can come together, yes or no? So this was the thing. Classification system for living organism have hence undergone several changes over time. Like over time evolution has happened, over time so many things has happened that now it has reached to a better point. Got it clear? Okay, clear. Fine. Yes. Okay. Chalo. Next. 1 second. Next paragraph. Okay. Next paragraph. Now, Organism, that is classification of organisms in plants. So beside gross morphology, here we did it till here. Yes, though plants and animals, okay, though plants and animal kingdom have been a constant under all different systems, the understanding of what groups and organism to be included, okay, organism to be included, these kingdoms have been changing. That is, the number, the nature of other kingdoms have also been understood differently by different scientists over time. So here they're not telling much anything, they're just trying to say that even though, like, continuous constant changes have been happening, where the phylums have changed, the kingdoms have changed, like they're trying to add someone here, someone there, like that kind of adding and add-ons were happening continuously. Okay, clear? Okay, now let's move on to the next most important paragraph. Now, students, see, in the NCERT, when you're learning the NCERT, you might be thinking that, oh no, why have they given it this way? They've not given a lot of it. It is because from the two kingdom classification, there is actually three kingdom classification, four kingdom classification, and then R.H. Whittaker's five kingdom classification come into place. But since it is not a part of your NCERT, we do not learn it because this is mainly for the CBSE students, right? Uh, this channel itself is for mainly for the board students. But if you are a NEET student, you know that I usually teach three kingdom, four kingdom classification. At least I tell you the scientist name. Okay, at least I tell you the scientist name. So here for your NCERT base, you do not need that. Okay, for your NCERT base, you do not need that. So the next ever classification that was considered, okay, very importantly, was R.H. Whittaker's classification. What was it? R.H. Whittaker. He came up with the most accepted classification. Most accepted classification. Now when I'm telling most accepted means what? Okay, that was the classification that was, agreed by everyone. Okay, agreed by everyone. Got it? Everyone were like, okay, wait a minute, actually this classification makes a lot of sense because he has gone into the depth of the classification. Okay, so what did he take into consideration? Now students, the biggest problem that all these other scientists faced was they did not look into the depth, right? One second. Okay, the main thing that other scientists did and RH, like other scientists did not do and RH Whittaker did was he went into the depth. Okay, he went into the depth. Now let's see what is the depth that he went into to understand the classification. So the first ever thing that he took into consideration was the cell type. Like, are you prokaryotes or are you eukaryotes was the first ever thing that was taken into consideration. Clear? Clear? I think, yeah. Okay. Hi, Akansha. So that was the thing. Second thing, cell wall was taken into consideration. That is, what are you made up of? If you have a cell wall, what are you made up of? Is it common for everyone or is it different? Okay, that one classification was done. Then nuclear membrane. Obviously, if you're doing prokaryotes and eukaryotes, you have to understand that you have to look into the nuclear membrane as well. Then we looked into the body organization. Are you cellular level? Are you tissue level? Are you organ or are you organ system? Okay. Then mode of nutrition, that is, are you autotrophic? Can you make your own food? If you can make your own food, what is the technique? If you're heterotrophic, then what is the technique that you're using? Like, are you saprophytic? Are you parasitic? All this was taken into consideration. Now if you look back, we saw Carl Linnaeus classification. What was the classification based on? Gross morphology. That is, you look like this, so I have categorized you, but I have not looked into the anatomy of it. Okay, so yes, so here students, cell type. Obviously, Monera was the primitive kingdom that had all the prokaryotes. Okay, all the prokaryotes were put into Monera. Then obviously from Protista, fungi, plantae and animalia, everything was U. That is eukaryotes. Okay. Now, cell wall in Monera was non-cellulosic polysaccharide plus amino acid complex, or you can also call it as peptidoglycan. Peptidoglycan, or you can also use the word, if you're confused, ma'am, I cannot remember polysaccharide plus amino sugar, then you can remember amino sugars. Okay, you can remember the word amino sugars. Clear? Fine. Yes. Now eukaryotes. Now here non-cellulosic polysaccharide plus amino acid, that is peptidoglycan, you can remember, or you will remember the word amino sugars. Now here Protista, they have written present in some, absent in some. That means some have cell wall, some do not have cell wall. Okay, some have cell wall, some do not have cell wall. So that is what here they are meaning by Protista. Now fungi, it is present with chitin. That is, the cell wall is made up of chitin. Now understand, fungi is like that one person in the class who does not get along with anyone. What to tell, you know, you people don't like the teacher's pet, no, or the person who spams everything, like goes and tells to the teacher that, you know, ma'am, she did not do the homework, she copied it from me. That kind of a person this fungi is. We try to put, or every botanist and zoologist try to put fungi in different place, but fungi was not getting along with everyone. So R.H. Whittaker was like, wait a minute, let's not take tension, let's give fungi a kingdom by itself. So fungi got a kingdom by itself. Okay, so cell wall is made up of chitin. Now plantate, do I have to tell, it is made up of cellulose. Okay, and animal, it's completely absent. Students, I'm teaching you in depth, so this is more than enough for your boards. Okay, even for NEET students almost is there, just that some extra points I'm not discussing here. Okay, I'm sorry, I have a cold, so I'll be might disturb you sometimes. Okay, now nuclear membrane. As soon as the word prokaryotes come, you know that the nuclear membrane is absent. That is the main reason. Then everywhere it is present. Okay, body organization. Students, as I told you, Monera is very primitive. You can imagine that it is going to be cellular level of organization. Protista also is going to be cellular level of organization, because understand, from Monera to Protista, it's not like a big jump. There are organisms, but they're not as primitive as Monera, but they're still primitive. Okay, so it's not that big of a jump also. Then here, when you come to fungi, you will notice that there are some multicellular, some loose tissues as well. Then in plantae, you will notice that there is tissues, organ. Animalia, there is tissues, organ, and organ system. All the categories are there. Okay, this you have to by heart. You don't have a choice. You have to by heart. Now, mode of nutrition, autotrophic. In autotrophic, please remember, here you have to learn something extra. Okay, you have to learn something extra. The extra is that, if I write it here, it's visible, no? Yes, in Monera, okay, in Monera, they have autotrophic. Okay, they are autotrophic. Students, if you are a student who is looking for notes, then you can definitely join my Telegram channel, that is Biology by Gopika, where you will get all the notes of my classes. Okay, yes, autotrophic students, two categories are there. Okay, two categories are there. Now, what are the two categories? If you look here, you will see that one is called as chemosynthetic. One is normal, photosynthetic. Okay, photosynthetic. Now, ma'am, what is chemosynthetic? I have learned photosynthetic from a very small age. Yes, chemosynthetic, students, it is actually what, you know, chemosynthesis, where we use, okay, the Monera uses chemicals to make their own food. To make own food. To make their own food. Got it? Now, photosynthesis means what? We use light, we use chlorophyll, and we make the own food. But here, what is going to happen? They are going to use different types of chemicals. Using that chemicals, they are going to make their own food. So here, the only thing that you have to remember very carefully is, under autotrophs, you have chemosynthetic and you have photosynthetic. Okay. Now here, you will notice that here heterotrophs also are there, parasitic, saprophytic and parasitic, nothing different. Here also autotrophism and heterotrophism is seen in Protista. Fungi also, you can see heterotrophism is the main thing, that under that you have saprophytes and parasites. Okay. Plantae, you can see autotrophism itself, photosynthetic. In Animalia, you will see heterotrophic, where you have holozoic, where we take the food, we digest it, and then the nutrition is absorbed. Okay, clear? Yes, and then we have saprophytic. So this box is the only box that you have to learn for RH Whittaker. You have to remember cell type, cell wall, nuclear membrane, body organization, mode of nutrition, and each of them: Monera, Protista, fungi, plantae, Animalia. In that, whatever is special, like Monera have peptidoglycan, it's a little special component. Fungi has chitin, special. Now, Monera shows autotrophism in two types: chemosynthetic and photosynthetic, special one. So like that, special ones always noted, because questions will come from the special one. They will not ask what is very common. Okay, what is very common. Students, actually, I'll just, 1 second. I'm just checking about the light. Yes. It's too bright. Okay, I'm not able to look at the screen. Yes, it's fine. Okay, here they're talking about RH Whittaker's classification. So let's just see that. Okay, I'm just going to check the light. After that, we'll start the class. Okay, we'll continue. Most of you haven't marked your attendance. Quickly mark your attendance as well. Okay, now R.H. Whittaker in 1969 proposed a five-kingdom classification. The kingdoms defined by him were Monera. Okay, one second. Monera, Protista, fungi, plantae, and animalia. Now, whatever box we learned, no, that same box they are going to talk about. Okay, the main criteria for classification used by him included cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. These were the things that were taken into consideration. Okay. Now, students, this topic, no, phylogenetic relationship was one of the click point. Okay, one of the click point of R.H. Whittaker, where they were like, wait a minute, if you have gone into the evolution, phylogenetic characters means what? You go back to the evolutionary relationship. Okay, you go back to the evolutionary history, okay, or relationship, anything you can remember. Evolutionary history. Now you tell me, we go back to the ancestors and we are like, okay, these are the things that has come through our ancestors, these were the traits that we see in this family. It cannot be wrong, right? Because we have gone back to the past and from there we are coming out, so it cannot be wrong. So it was confirmed that whatever R.H. Whittaker has done is not been done by any other scientist, and because of which it is going to be the most approved one. It is going to be the most approved one. Is this clear? Okay. So, table 2.1, that is the table we just did, gives a comparative account. Okay, comparative account of different characteristics of five kingdom. The three domain system has been proposed and divided to kingdom Monera into two domains. Now the problem is, a student normally reading NCERT will not understand what this line is talking about. I'll tell you what this line is talking about. There was a the scientist called as Carl Woese. Okay, Carl Woese. He came up with six kingdom classification. Obviously, after five kingdom, he came up with six kingdom classification. Now, what is this classification? Six kingdom classification was like, okay, let's do one thing. Let's make it into three domains. Okay, three domains and six kingdom. This was the plan. Now, what is this three domains? Domains are above kingdoms. Okay, so he categorized the Monera's two parts into Archae, that is all the Archaebacteria. Okay, Eubacteria and eukaryotes. So this became three domains. Yes or no? This became three domains. This three domains he categorized into six kingdoms. Okay, how is that possible? Under Archaea, obviously Archaebacteria came. Under Eubacteria came. Under eukaryotes, he put all four of them. He put Protista. He put fungi. He put plantae and animalia. So this became how many students? Under Archaebacteria, under Eubacteria, under eukaryotes, four of them. So a total of six kingdom classification. So this is what they're talking about. Three domain, six kingdom classification. So if you read by yourself, you have no chance of understanding this. That is why it's important for you to learn from a teacher. Okay, done? Yes or no? Done, right? Okay, I'm just checking if the live is also happening smoothly. Yes, it's happening smoothly. Okay, so that was clear. Now we will proceed to the next paragraph. Now let us look at this five kingdom classification to understand the issues and consideration. What are the issues and consideration that influence the classification system? That is five kingdom classification. Also, it's slightly here and there issues with it. So let's see which were the issues. Earlier classification system included bacteria, blue-green algae, fungi, mosses and ferns, gymnosperms, angiosperms under plants. Whose classification they are talking about here? They are talking about Carl Linnaeus classification. They're talking about Carl Linnaeus classification. What he did, he put bacteria, he put blue-green algae, he put fungi, he put mosses, ferns, gymnosperm, angiosperm, everything into, okay, one classification. Okay, one classification. Got it? And he put everything into plants. Now, the character that unified this whole kingdom was that all organism included had a cell wall in their cells. This was the one reason why he put everything together. Okay, he put everything together was because cell wall was there in the cells. Okay, this placed together groups which widely differed in other characteristics. So it brought together the prokaryotic bacteria and the blue green algae, which was the cyanobacteria, and other groups which were eukaryotes like your fungi. Okay, it also grouped together the unicellular organisms and multicellular organisms. One, say for example, chlamydomonas. Okay, chlamydomonas and spirogyra were placed together under algae. Okay, the classification did not differentiate between heterotrophs and so here also some issues were faced. The issues were where that first issues were that all the cells which had cell wall was put together. Okay, be it angiosperm, gymnosperms, ferns, mosses, primitive to high class plants were put together. That was the problem, but later on they tried to rectify each of the issues. Okay, rectify each of the issues. One second. Yes, they try to rectify each of the issues. Now, for example, chlamydomonas and spirogyra were placed together. That was an issue. Then, let's look at the further one. Okay, heterotrophic. The classification did not differentiate between heterotrophs. Right. Yes. Okay. Yes, the classification, see here, were considered the fungi were placed into a separate kingdom, kingdom fungi. All prokaryotes organism were grouped together under kingdom Monera. All the prokaryotes, okay, all the prokaryotes were put under the category of kingdom Monera, and unicellular eukaryotic organism were placed in kingdom Protista. So this was the classification key. Okay, let's do one thing. Let's put all the prokaryotes. Okay, let's put all the prokaryotes into one place. Okay, let's put all the prokaryotes into one place. Next thing what we will do, we will put all the unicellular eukaryotes into Protista. This was the plan. Looks fine, no? That is Monera. So if I'm taking Monera, I'm going to put all the unicellular. Okay, I'm going to put unicellular and all the prokaryotes. Okay, all the unicellular and prokaryotes. Then Protista, if I'm taking Protista, I will put all the unicellular and eukaryotes. Obviously, in eukaryotes also there will be some unicellular, no? That also had to come into place. Now, kingdom Protista has brought together Chlamydomonas, Chlorella, earlier which was placed in algae within plants and both having cell walls, with Paramecium and Amoeba which were earlier placed in animal kingdom. Now, students, earlier, the word they are using is for your two kingdom classification. Don't get it confused. What they are trying to say is before these people were put together, now we brought them into one place depending on this two condition, that if they're unicellular, if they're prokaryotes, they will go to Monera. If they're unicellular and eukaryotes, they will go to Protista. Simple classification was done. Okay, simple classification was done. So here you can see this examples you have to remember: Chlamydomonas, Chlorella, okay, and Paramecium amoeba, okay, which were earlier placed in the animal kingdom, which lack cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. Okay, this happened because the criteria for classification changed. So as in time came, classification started getting modified a little, like that, wait a minute, instead of doing the so complicated classification, let's do one thing. Whatever is unicellular prokaryotes, let's put them into one era. Whatever is unicellular eukaryotes, let us put them into Protista. This way what is going to happen, lot of confusions won't come. Okay, so this kind of changes will take place in the future too, depending. So that they're telling that now because you're studying this does not mean this is the end of it. In future also such changes can come, and depending on the changes, if they feel that why is this organism placed here, it does not make sense, then this organism can be shifted. Is this clear so far? Okay, so far good. Yes, let's take a water break. Okay, drink water all of you. Yes. Okay. Now, over time that you leave, over time, an attempt has been made, okay, has been made to evolve a classification which reflect not only on the morphology, physiology, and reproductive similarities, but also on the phylogenetic, that is based on the evolutionary relationship. This is what I told you in the starting of the class, that R.H. Whittaker's classification could not go wrong because R.H. Whittaker did the classification based on phylogeny. Phylogeny means what? Evolution, right? Evolution cannot go wrong. All of you have learned evolution chapter, no? Evolutionary relationships cannot go wrong. Okay, evolutionary relationships cannot go wrong, even though we do some, like sometimes what happens, no, we just look at the organism, we are like, okay, this is how it looks like, this is where we have to categorize it. Sometimes mere morphology, sometimes even anatomy, sometimes even reproduction can go wrong, but evolution will not go wrong. Okay, that is like where you go to the past, you find out the loopholes, and then you find, like, get to a categorization or get to a place. That is a different story by itself. Okay, now in this chapter we will study about kingdom Monera, Protista, fungi, and Plantae and Animalia. By itself you have two chapters called as plant kingdom. Okay, plant kingdom and animal kingdom. Okay, done. Yes, yes, done. Okay, so plant kingdom and animal kingdom, commonly referred to as plant and animal kingdom respectively, will be dealt separately in chapter 3 and 4. Done. Okay, this is the whole thing will be dealt in chapter 3 and 4, that is plant kingdom, animal kingdom, where you have to study more about it. Okay, you have to study more about it. Clear? Okay, done. Yes. Okay, we're going to start with kingdom Monera. Okay, kingdom Monera. I think placement is fine. One second, please. Yes. Students, voice is fine, huh? Some students are telling. Voice is fine only, no? Okay. Yes. Chalo, let's continue the class. Okay, so botany classes also will start happening depending on you people's request. You can tell which chapter to start. Then we will be starting those chapters. Okay, chalo, we'll do Kingdom Monera. Okay, Kingdom Monera. Now, students, as I told you, Kingdom Monera, when you remember, you have to remember one thing mainly, that these people are unicellular. Okay, unicellular, and they are okay, they are prokaryotic. And then I also told you, you have to remember that these people are autotrophic, and in autotrophism they show two different things. That is, one, they are chemosynthetic. Okay, one, they are photosynthetic. That is, one, they use light, one, they use chemicals. Okay, now bacteria, very important, listen to me carefully. Bacteria are sole members of kingdom Monera. That means what? You have made a group. Okay, you have made a group, and it is called as girl squad. Who will be majorly in the group? All the girls will be there. No? Same way, in this Monera, the main members, or the whole people who are constituting to this, is your bacteria. Okay, they are most abundant microorganism. Yes or no? Now if you take my hand and see, you will see millions of bacteria in my hand. Same way in this pen, anything. Okay, this hairband, whatever you touch right now, there is a lot of bacteria, but they are not that harmful. Okay, they are not that harmful. Clear or not? What is happening? One second, students. Okay, done. Okay, yes. They are most abundant microorganism. Bacteria occurs everywhere. Okay, bacteria occurs everywhere. Hundreds of bacteria present in a handful of soil. Okay, a handful of soil. So many bacteria are there. Okay, so many bacteria are there in a handful of soil. Yes or no? Agreed or not? Now, if any person takes a handful of soil and checks it, they'll be like, oh my god, so many bacteria. From where did this come? Right? They also live in extreme habitat. This is what makes bacteria important, students. It's not about it is there in handful of soil or not. That is not important. What is important is that even at very extreme conditions, they'll be able to survive, like hot springs, deserts, snow, deep oceans, where every, like, in a sense, even few organisms are not able to survive. No, there also these people will be able to survive. Got it? Okay. Yes. Okay. So where very few life organisms can survive, many of them live in or on other organisms as parasites as well. So where and all do they live? They live in many places. Okay, they live in many places. Got it? So bacteria are grouped under 4 categories based on what? Based on their shape also. Now let's look at the shape. The first ever shape is spherical shape or coccus. Okay, spherical or coccus shape. Then we have rod shaped or bacillus shaped. So this is the most common one you see. Okay, my drawing only is like that shape. Then we have vibrium or comma shape. Then we have spiral or spirulum shape. Okay, spiral or spirulum shape. Now students, what are they talking about? 4 categories of bacteria. Okay, based on the shape. Based on shape. Okay, based on shape, we have very important. First one is called coccus, second one is called bacillus, third one is called vibrio. See, first time when you listen to all this you might get heart attack, but later on you will be very versed in this chapter. Okay, like well versed in this chapter, not versed, well versed in this chapter. Okay, well versed in this chapter. Okay, that is coccus, bacillus, vibrio, spirillum. Now coccus means what? Spherical in shape. Okay, coccus means what? Spherical in shape. So it will be like this. Okay, like this. Coccus. Bacillus means what? Rod shaped. Okay, bacillus means rod shaped. Okay, vibrio means comma shaped. Vibrio means comma shaped. Spirillum means spiral shape, like this, like this. Okay, like this, zigzag, it will be going spiralum shape. So depending on the shape, we have categorized bacteria into 4 categories. Is it clear? Okay, chalo, let's move on. Yes, this is the diagram. Okay, this is the diagram. Look at the diagram. Look at the diagram. In your NCERT it is a beautiful brown color diagram. In mine it is black and white. Okay, so this is spherical. For coccus I'll tell spherical. Okay, this is bacillus, that is rod shaped. Spirilla, that is spiral. And vibrio, comma shaped. When you write in boards, don't write comma, the other comma, write one more M. Okay, yes, done, all of you. One second, students. Yes, done, all of you. Yes, clear, very good. Okay, very good. Now let's continue. Okay, I have to move this. Okay, see, now we are going to start. Okay, kingdom Monera. Now we understood that the sole members of kingdom Monera is bacteria, and these people are present wherever, you know, other organism cannot survive. All those places these people are present. Okay, all those places these people are present. Now here, now though the bacterial structure is very simple, okay, though the bacterial structure is very simple, They are very complex in behavior compared to many other organisms. Bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophs or chemosynthetic autotrophs. The vast variety of bacteria are heterotrophs, that is, they depend on other organisms, especially on dead organic matter for food. So we think no, bacteria maybe majority is autotroph, but majority are heterotrophs, very few are autotrophs, because they need to have the pigment. If they do not have the pigment, how will they do any photosynthesis, right? If they don't, like, other ways they use the chemicals to make their own food. Okay, got it? Chalo. First one, Archaebacteria. Archaebacteria. First is category of Archaebacteria. You will be like, ma'am, you were teaching Monera. How did it come to Archaebacteria? Archaebacteria comes under Monera. Okay, class is like this going, students. Okay, kingdom, Monera. Okay, divided into two groups. One is called as Archaebacteria. Archaebacteria and Eubacteria. These two things only, Carl Woos made it into a domain by itself, telling RK and U will become a domain by itself. Under that, bacteria will come. Okay, but then that is six kingdom classification, which is not very widely accepted. Now, these bacteria are special since they live in some of the most harsh habitats. Okay, such as extreme salty areas like halophils. Okay, hot springs called as thermoacidophils, marshy areas called as methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for the survival in extreme conditions. Okay, methanogens are present in the gut of several ruminant animals such as cows and buffaloes. That is the reason why they are able to form the gobar, right? Biogas they are able to form because this cow dung has so much of methane, and that will be used, which you learn in microbes chapter next year, that they will use that. Okay, and they are responsible for the production of methane, that is biogas, from the dung of the animals. So what are they trying to say? I'll write it for you. That is, Archaebacteria, main thing is they are able to withstand any extreme conditions like salty conditions. Okay, I'll just do one thing. I'll write only that. Okay, salty conditions like halophiles, extreme salty conditions. Okay, extreme salty conditions like halophiles. Next one is thermoacidophils, that is extreme pH, extreme acidic condition, extreme heat. They're like, hey, this is an all small things for me, I can stand in any climate. Okay, they're like, I can stand in any kind of climate. Okay, so that is what they're talking about. I can stand in any kind of climate, don't worry, I'll do it. Okay, Archaebacteria, that is halophiles, thermoacidophils, and then even in marshy areas. Okay, marshy areas and in the gut of ruminant animals, you have methanogens. I'm writing the name. Okay, so Archaebacteria is divided into three categories where you have um, okay, three categories where you have halophils, which are extremely salty conditions. You have thermoacidophils, extreme heat condition plus acidic condition. Okay, and you have methanogens, which is extreme marshy areas, but they are also present in the cattles. Okay, methane, a lot of methane will come out through this cattles. Clear? So Archaebacteria's classification we saw. Main thing is, students, why are they able to be in such an extreme condition? That also we have to think. Chuma we're telling that okay, this is there here, this is here. We should also think why they are able to withstand. What is making them so special that other organisms are not able to withstand? The reason for Archaebacteria, I'm writing it everywhere. Okay, for the reason for Archaebacteria to be able to withstand or go through this extreme condition, is main reason is because of their false cell wall. Okay, false, that is cell wall, that is called as pseudomurin. So you might get confused, ma'am, you never told this word before. So remember I told you peptidoglycan. Peptidoglycan, I use the word. That peptidoglycan's other word is murin. Peptido's other word is murin. Now this Archaebacteria have false murin. false murin, obviously false word can be replaced by pseudo. So I'll call it pseudo murin. So this is the reason why they are able to withstand extreme conditions. Okay, withstand extreme conditions. Clear or not? Clear or not? Okay. So much ad is coming. So much ad about milk is coming. I don't know why. Like to give you energy. Okay, handwriting is visible, no? Withstand extreme conditions. Got it? Yes. We will move on. Eubacteria or true bacteria. You always remember, no? U means always true. So there are thousands of different eubacterias or true bacterias. They are characterized by the presence of a rigid cell wall, if motile, a flagellum, and the cyanobacteria, also referred to as blue-green algae or BGA we tell, have chlorophyll A similar to that of green plants and are photosynthetic autotrophs. You'll be like, ma'am, if we did not understand anything, let's go one by one. Don't take tension. See, look here. First point first. They are telling you bacteria is true bacteria. Students, I want even your NCERT to look exactly like this, where every corner and side, no, you're writing the notes and keeping, you are underlining the important words. Otherwise, your NCERT will look new, right? It will look so new that no, everyone will be like wanting to buy it. But that's not the goal, okay? You have to use your NCERT because NCERT is the only thing that is going to help you in your boards as well as your competitive exam. Now, if you're a student who is going to write for NEET, you know very well that we keep telling NCERT, NCERT, NCERT because every question is framed from NCERT. And there is a knack to understand NCERT. That is why we are doing this NCERT line by line. Okay, it's actually more beneficial for the students to do NCERT line by line because I am reading it and I am explaining every line. So you should take maximum benefit out of it and you should be able to write the notes in the side as I'm telling. Okay, so here, see, now I've taken the snip here, I've put the space here, so I will be able to write as much as possible. So eubacteria or true bacteria, they're telling that the thousands are present. They are characterized by what? Why am I calling it true bacteria? Because of a rigid cell wall. Okay, rigid cell wall. Got it? And if motile, like if they are able to move, they'll have a flagellum for sure. Now, one of the most common example is cyanobacteria. We will write it in blue itself. Cyanobacteria, which is otherwise called as BGA, blue-green algae. Okay, BGA or blue-green algae. The cyanobacteria, students, here they're talking about cyanobacteria only. They are unicellular. Unicellular. Okay, they can be colonial. Colonial means what? Like one same set of people stay in a same area, we will call it a colony, right? We will call it a colony, like teachers colony. That means all the teachers have stayed there before, so name came like the teachers colony, like that. Okay, filamentous freshwater colonies. Okay, they form colonies. They can be found everywhere. They can be seen in freshwater, they can be seen in marine water, they can also be seen in terrestrial. Okay, now very important, if a colony is there, we will make sure to build a fence around the colony. Yes or no? So that no one other, random people start coming in and telling that okay, when we want to be a part of the colony. So same way here also, imagine these are the colonies. Okay, this is one colony, this is one colony. Okay, this is one colony, this is another colony, this is another colony. Like this colonies are there. Okay, to protect this colony, which color do we use? We'll use yellow. Okay, there is a mucilaginous covering. Okay, mucilaginous covering. So like this we'll be able to cover. Got it? There's a mucilaginous covering, or we also can call it as a gelatinous covering. Okay, gelatinous covering, or other word is mucilaginous covering. Why is this covering, students? Obviously same thing, no? I build a home, I'll definitely build a fence. So to protect this colonies, we have a gelatinous sheath. See here, look here, surrounded by a gelatinous sheath. They often form blooms in polluted water bodies. Now, how many of you have gone to village? Like when you're going to village, when you're traveling in train or bus, you'll notice that there is a lake which is completely green in color, and you'll be like, it looks beautiful. But what has happened? Algal bloom has happened. That is, the entire algae have multiplicated, rapid multiplication has happened, and they have covered the first layer of the water. Okay, first layer of the water is completely covered by the green color. Now what is going to happen? They will consume all the dissolved oxygen. They'll also make sure that there is no sunlight that is going downwards. They'll make sure that there is no passage of sunlight that is going, and because of which they have already taken all the oxygen. Now they'll not also not give the sunlight. Now what is going to happen? The plants will not be able to make the food, the ones which are down, and the aquatic organisms, all of them will die. Okay, so that is called as algal bloom. Now see, some of these organisms can fix atmospheric nitrogen. It's a very very important characteristic. Like if you are writing for exam, this point will fetch you all the mark. Okay, this point will fetch you most of the marks. Clear? Voice disturbance might come. Yeah. Yes, ready? Okay, so what is going to happen? They have now in this colony. Okay, let's in this colony itself, students, let me take another color. Okay, let's take this dark green, and in this, I think it won't be visible. No, is it visible? One second. This one cell. Okay, one cell. Yes, can you see? One second, let me just see if you're able to see. This one cell is called as a heterocyst cell. What is the cell called as? Heterocyst. Heterocyst. Heterocyst means what? The cell that can fix nitrogen, right? The cell that can fix nitrogen. Can you see a light dark green I have drawn? Yeah, okay. That cell is called as a heterocyst. That is, they can fix nitrogen. Done? Okay, okay. Yes, they can fix nitrogen. Okay, I'm going to just check my voice. Okay, I'm going to check my voice. That is, they can fix nitrogen. Yes, voice is audible, no? Heterocyst. So example, the ones which have this heterocyst cells are Annabina. Students don't write Annabella. Okay, Annabina and Nostoc. Nostoc. Okay, Annabina and Nostoc. And now we are going to chemosynthetic. We have been talking about it so much and we have not gone into it, no? So look here. Chemosynthetic autotrophic bacteria oxidizes various inorganic substances such as nitrates, nitrites, ammonia, and are used to release energy for ATP production. Main job is what? I make food for ATP, right? So what these bacteria are going to do? They are going to take the nitrites, nitrates, ammonia, and they're going to utilize it to release ATP or to form ATP. This kind of synthesis is called chemosynthetic autotrophs. That means they can make their own food but with the help of chemicals. Okay, and they play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. What and all? So let's use all the chemistry knowledge that we have. Okay, let's use the chemistry knowledge. They're telling nitrogen, phosphorus, iron, Fe, and sulfur. Correct? Okay, NPFES. Okay, NPFES is what is be recycled because of these people. So very simple. What and all did we go through? I think I have made a chart here. See here, this is what I told you, colonies. Okay. Now, heterotrophic bacteria are most abundant in nature. The majority are important decomposers. This is an important question. Okay. Important decomposers. Don't forget this. Okay. Don't forget this. Important decomposers. Many of them have many of them have significant impact on human affairs. They are helpful in making lactobacillus. All of you remember in small age also we have studied, no, lactobacillus will convert milk to curd. So they're talking about lactobacillus. Students, bacillus means what? Rod shaped bacteria. So you have to keep connecting with each other. Okay, chumma studying by hearting will not fetch you biology marks. You have to connect with each other. The moment you listen lactobacillus, you should be like, okay, wait a minute. Bacillus means ma'am told it is rod shaped. And lactobacillus is actually a useful bacteria because it is converting milk to curd. Okay. Penicillium, many things are there. Okay. Then we have leuconostoc that ferments your dosa and idli. Got it? Yes. Okay. Done. Now we will continue. Yes, so they have told that bacteria are not always bad. You do not have to think that bacteria are always bad. They are also actually doing some good things for us, right? Now look here. Some are pathogens causing damage. There are bacteria which definitely cause damage, right? That is the reason why we tell you should always wash your hands when you come out from outside. When the kids are playing, we tell no, if you're going outside, please come back and wash your hands. That is because there are bacteria that actually causes problem. There are also bacteria that affects the crops, farm animals, pets, and all that. No doubt in that. But there are also a lot of good bacteria which you study in microbes chapter. Okay. Now here you can see the diseases caused, like typhoid, tetanus, cankers, and are known diseases of different bacteria. Bacteria reproduce mainly by fission. Now this is the important part, that is about the reproduction of bacteria. Still now what did we talk? We spoke everything, like we were like, okay, bacteria is like this, bacteria is like this. Now we are talking about how the reproduction of bacteria is happening. That is, how are they actually reproducing? So students, if I draw the bacteria, okay, this is one diagram given in your NCERT as well. Similar diagram, okay, it can't be the same, but yeah, similar diagram. Okay, yeah. Now if the reproduction has to happen, it will happen with the help of fission, right? Fission will happen. This will become a complete individual, this will become a complete individual, so it will happen, fission will happen. That is the first thing they have told. Now, under unfavorable condition, many times favorable condition, unfavorable condition comes. So, sometimes under unfavorable condition, they produce spores. So, how do they do? Most of them do, okay, students, because for their survival rate, they need to form spores. Otherwise, they're not going to survive. So, during the condition gets unfavorable, they'll form spores. They also produce by a sort of sexual reproduction. Now, all of you tell me, from the time bacteria we know, okay, from our ancestors time we talk a lot of a bacteria, lot about bacteria. From that time is the bacteria same? No modifications have happened, no improvement has happened, or what? Lot of improvements have happened, right? Lot of improvements have happened, but how did the improvements happen? The bacteria, on the long run, we understood that they're not doing sexually anything, okay, sexually anything, but they have got categorized. How is that possible? Right, they have got categorized, and how is that possible? That is when people told that, wait a minute, bacteria actually does sexual reproduction, but not in a full-fledged way like how we know, but they does do something called as transformation, which you will be studying in molecular basis of inheritance chapter under Griffith's experiment. Okay, transformation or transforming principle you study. What happens there is an S strain, okay, which is actually virulent means disease causing. So this scientist heat killed the S strain, and he injected into the mice. The mice lived. Then he used R strain. R strain is what, student? It is non-virulent. Okay, Pavam it is. S strain is the villain. So S strain is virulent. The scientist Griffith, no, he heat killed the S strain and he injected into the mice. Mice was happy, did not die. So what he thought, he was like, okay, wait, two harmless strains, I'm going to mix it. That is, heat killed S strain and R strain. He tried to mix it. Suddenly he noticed that the mice were supposed to live. Yes or no? Because when I individually use this, that fellow lived. When I individually use this also, that fellow lived. But when I cross this both, okay, when I cross the heat killed, this you remember, okay, heat killed S strain and R strain, the mice died. Then this Griffith was like, wait a minute, there is something that got transformed from this S strain to R strain, and he called it as the transforming principle. Now, transforming principle means what? DNA got transformed from the S strain to the R strain. So, they are telling that bacteria actually does sexual reproduction in the form of transformation, conduction, all that. Okay, yeah, with the form of all this. Okay, transduction. Okay, transduction. Okay, conjugation. These are the technique, not conduction. Okay, conjugation, transduction, transformation are the techniques by which the bacteria actually does sexual reproduction. But do not think that it is happening at a high level. It's happening at a kuti level only. Okay, done. Okay, okay, done. Now, this one is very important, students. The first word that is lacking, I'm going to write the word. It is mycoplasma. Okay, mycoplasma. So mycoplasma completely lack cell wall, and they are the smallest living cells known, and cannot survive without oxygen. Okay, sorry, and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. That is, the smallest cells that we know for so far is mycoplasma. Mycoplasma is the completely lacks cell wall. Very, very important. Okay, completely lacks cell wall, and they are the smallest what living cell we know. Okay, smallest living cell that I know. Done. Okay, smallest living cell I know. Now, one second. Okay, after this we'll take a cutie break. Okay, after this we'll take a cutie break, and we'll come back and we'll finish Protista. Okay, now whatever we studied we are going to write. Okay, give me 1 minute. Yes, very good, Shubham. They are very small, right? They're very, very small. Yes, ready? Okay, whatever we studied so far, we are going to write. We are not going to write about classifications. We are going to write about Monera. Okay, we are going to write about Monera because classifications will take too much time. So we'll write about Monera. So here we'll write Monera. What did we learn about Monera? First thing I know about Monera is that the sole members of Monera, okay, sole members of Monera are bacteria. Okay, are bacteria. Okay, got it? Sole members are bacteria. Next thing I know is they'll be able to withstand extreme conditions. Okay, they can survive in extreme conditions. This is what I learned about Monera. Then what did I learn, students? Monera is present everywhere, right? It is everywhere, so I will call it the word cosmopolitan. Cosmopolitan, that means everywhere. Okay, now I got to know that Monera is classified into two categories. Monera is classified into two categories. Okay, Archaebacteria, Archaebacteria and Eubacteria. Archaebacteria and Eubacteria. In that, we'll start with Archaebacteria. First, I got to know that Archaebacteria have false cell wall. Yes or no? Yes, cosmopolitan. Okay, they have false cell wall. False cell wall means what? They do not have the proper peptidoglycan that I spoke in Monera. They have pseudomurin. I told you peptidoglycan can also be called as murin, so I will call it pseudomurin, which allows them to withstand extreme conditions. Okay, and then Archaebacteria is categorized into three categories depending on where they'll be able to survive, like halophils, extreme salty condition, thermoacidophils, that is extreme acidic condition or extreme heat condition, and the last one is methanogens. Okay, methanogens. Got it? Now, what do I know about each of them? I'm using different colors so you'll remember. Okay, pseudomurin halophiles are extreme salty condition. Okay, salty condition. Thermoacidophils extremely acidic plus heat. Okay, extreme heat. And methanogens are present in ruminant animals like your cattle, which will help them. Okay, ruminant animals, which will help them to form biogas. Okay, biogas. Done? Did I miss something? Did I miss something? Yes. Now, from there, we move on to Archaebacteria. Done. Okay, Archaebacteria done. Next, we go to Eubacteria. Okay, Eubacteria or true bacteria. They are called as true bacteria. I'm writing it here again. True bacteria. Okay, true bacteria. Now, other than true bacteria, what do I know? They have cyanobacteria in them. Okay, Archaebacteria consist of cyanobacteria, which is called as BGA. And the most important thing is the cyanobacteria has chlorophyll A exactly like green plants. Students, my arrow marks you have to understand, okay? Green plants. Got it? Full flight takeoff handwriting is going, can't do anything. Okay, chlorophyll A, you can keep a sheet. Yeah, okay, chlorophyll A. Yes, chlorophyll A, green plants. Then we got to know that they are found, can be colonial, they can be present in colonies, or they can be filamentous. Okay, they can be filamentous. And if they are, and they are found in terrestrial, aquatic, all the places. If they are found colonial, they will have a covering. Okay, they will have a covering called as the gelatinous sheath. Okay, gelatinous. Oh, okay, they have a gelatinous sheath. Got it? Now that is for the protection. Okay, and students, what is there in this colonies? There is a presence of a cell called as the heterocyst cells. Okay, heterocyst cells, which can actually do nitrogen fixation. Okay, which can actually do nitrogen fixation. Got it? Nitrogen fixation. Now, not only that, the examples were Anabina and Nostoc, which I'm not writing it there. Then reproduction in bacteria. Okay, we'll use another color so you understand it's a different point by itself. Okay, reproduction in bacteria is by fission, and sexual reproduction is by transformation. Done? Okay, sexual reproduction is by transformation. This is what we studied. Then obviously you have mycoplasma. Okay, mycoplasma, I'm writing it in the center. That is lac cell wall, and a special cell it is. Okay, mycoplasma, special one. So we learned Monera. Okay, we learned Archaebacteria, we learned Eubacteria. Done. Okay, again bacteria are classified based on shape and all that is there, which we cannot fit it into the same screen, right? We cannot fit everything to one slide. So this is what we have done so far. Okay, next we will start with Protista. Before that we'll take a kuti break. Okay, we'll take a kuti break. All of you recharge yourself and come. So we will resume back at 3:34. No, yes, 35. Okay, we'll resume back by 3:50. Okay, 3:50, all of you have your lunch and come. So we'll resume back by this time. Okay, done? Yes. Okay, students? Okay. Hello students, I'm back. Okay, yes, I hope I'm audible. Okay, there's no problem. Awesome. So we're going to do Kingdom Protista. We just finished Kingdom Monera and we revised Kingdom Monera as well. Now we will be doing Kingdom Protista. Okay. Today's class we are going to do Kingdom Protista as well. So all single cell eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. Right. Sometimes what happens is when we are trying to categorize something, sometimes we will not know the boundaries for this because they might be a connection between something, they might be a connecting link between something. So when this situations come, what happen is we are not able to set proper boundaries, right? Proper. Now you cannot set a boundary with your best friend because you already know he or she is your best friend and you have been for years. Now you cannot set boundaries saying that don't take my phone, don't come to my home, don't touch my bed. All this you cannot set because you know that they are connected to you and now you cannot make that difference. So what may be a photosynthetic protestant to one biologist may be a plant to another. This was one of the problems also that they noticed, that maybe I am thinking that it is a photosynthesis, like it is a photosynthetic protista. Okay, I am thinking that it is a photosynthetic protista, but at the same time, Shubham is thinking that no, it is actually a plant because it is able to do autotrophism. I consider it as a plant. Anything that is able to make its own food, I consider it as a plant. So like this things were there. Okay, in this book we include cryophytes, dinoflagellates, euglenoids, slime molds and protozoans under Protista. So under Protista we have 5 people coming into place. Okay, coming into place. That is, first one we have cryophytes, second one we have dinoflagellate, third one we have euglenoids, fourth one we have slime mold, fifth one we have protozoans. So these 5 people categorize and fall under protozoans. Okay, clear? Yes. Okay, I am just going to check my voice. Okay, that is, first one we have crime. Okay, done. Okay, slime molds and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with others dealing with plants, animals, and fungi. So what did I say? I told that they are going to be a link. So Protista, students, actually is a link. Okay, Protista is actually a link between plants, animals, and fungi. Now, not just link, okay? Each of them will have a category. Like example, this first 3 people, no, cryophytes, dinoflagellates, euglenoids are plants. Okay, they are plant-like. That is because they have chlorophyll, they have pigments, and they behave very similar to plant. So CDE will fall under, okay, CDE will fall under plant-like character. Then you have slime mold, which is actually acting like a fungi. Okay, which is acting like a fungi. Then you have protozoan, which is going to do the locomotion and movement. So we will consider it as under animal. Okay, so that means in this category Protista, where we study 5 types, in this 5 types we notice that there is a connection between animals, plants and fungi. Okay, now being eukaryote, the protestant cell body contains a well-defined nucleus, we know that, and other membrane-bound organelles. Some have flagella or cilia. Protista reproduce asexually and sexually by the process of involving cell fusion and zygote formation. So these two things also are important for you to remember, that they also do cell fusion, okay, and zygote formation. Okay, zygote formation. Got it? So Protista is actually a link between plants, animals and fungi, which you have to not forget at all because this question has been asked multiple times. Now let's take cryophytes. Okay, let's take cryophytes and let's try to understand what they're talking about. Now students, chrysophytes you can tell, or you can tell cryophytes, anyone you can tell. This group includes, what do they include? They include diatoms and golden algae. Okay, diatoms and golden algae. I'm actually using gold color. They are found in fresh water as well as marine water. So first thing we learned was these people have diatoms. Okay, these people are diatoms and also golden algae. Okay, golden algae which are called as desmids, which are called as desmids. They are found in freshwater as well as in marine environment. They are microscopic, okay, and float passively in water like planktons, okay, like zooplanktons, no, they will move passively. Now most of them are photosynthetic in diatoms. The cell wall, this is the reason they are mostly photosynthetic. This is the reason why we call them a plant like, okay, we call them plant like is because of this one reason, that they are photosynthetic. Okay, in diatoms the cell walls form two overlapping shells. Students, very important in this whole protista, no, you have to remember about the cell wall. That is the most important thing. That is, the cell wall is overlapping, one shell like this, okay, one shell like this, and one shell like this. So this looks like a soap box, yes or no? Before, you remember, we used to have the soap box where the top was there, the down was there. I cannot find it here. So it looked like a soap box, okay. So two overlapping. So the most important thing in this chapter is you have to, I mean in this protista, is you have to know about the cell wall. So in first cryophyte, cell wall is two overlapping shells which fit together in the form of a soap box. Got it? In the form of a soap box. Don't even forget this. Very, 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 very important. Okay. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind a large amount of cell wall deposit in the seabed in their habitat. This accumulations after billions of years are called as diatomaceous earth. So they can ask you the definition for diatomaceous earth. As you are a board student, they might ask you this question, that what is diatomaceous earth. So what you have to tell, very simple, these overlapping shells, no, they are made up of silica. So imagine, let's take this pink dots only, okay. This is the habitat that they're leaving. This is the seabed. Wow, what a beautiful wave have I drawn. Okay, so beautiful wave has come. Now, after many years, students, this overlap, the cell wall has been completely falling. Okay, falling, and they have made a seabed out of it. They have formed a bed full of these things. Okay, now after billions of years, I will call this as diatomaceous earth. Why? Because this diatom cell wall is made up of silica which is not easily destructible, and because of which I will be able to use this diatomaceous earth, okay, diatomaceous earth, later for other purposes like polishing, filtration of oils, etc. So these people are the chief producers in the ocean. Who? Diatoms. We are not talking about desmids here. Mainly we are focusing about diatoms. So what and all did we learn now? Right now we understood that cryophytes have diatoms and desmids under them. Main thing is they are photosynthetic. They are found in marine environment. They are microscopic, floats passively as plankton. This questions and all. See, if they ask you general characteristics of cryophytes, you can write it. Otherwise, you do not have to actually take so much tension about this. Okay. Now they are microscopic and floats passively in water current. Okay. Most of these plants are photosynthetic. Very important. In diatoms, the cell walls are overlapping. Okay, they're overlapping to form a soap box like structure, and this overlapping shells are made up of silica, and this silica over billions of years have settled at the sea bottom or settled at the habitat, and later will be called as the diatomaceous earth, which can be later used for polishing and filtration, and this will be extracted. Okay, this will be extracted. Got it? This is the whole recap of your cryophytes, and this is the reason, one of the reasons why we put cryophytes under plant. Okay, cryophytes under plants. Done? Okay. Yes. 1 second, students. Give me 1 minute. I just want to see if the lighting is fine. Okay. Just let me know. Let me check as well. Because only if I see, I'll be able to know how the lighting is. Okay. Yeah, I think it's fine, right? Okay. So students, yes. What are we talking about? Diatomaceous earth. Then, ma'am, chrysophytes perfectly understood. Okay. Now we will move on to dinoflagellates. From the term itself, you understand. Dino means two, right? Dino means two. So we will use some other color. Dinoflagellates. These organisms are mostly marine and photosynthetic. See, this is the reason why we categorize the first three people under plants. Main reason is because of their photosynthetic nature. Okay. Now they appear yellow, green, brown, blue, or red in color. My god, how many colors do they appear into? Yellow, okay, blue, green, brown, red. Five colors, okay. Y, G, B, B, R. Okay, I remember like this. YGBBR. Okay, YGBBR is yellow, blue, green. I mean yellow, green, blue, brown, and red. This many colors depending on the main pigment. Depending on which pigment they have more, they will have some different types of colors. Okay, now the cell wall has. See, I told you, cell wall is the main topic in Protista. I don't know how many teachers would have told you this. Cell wall is only thing that you have to focus in Protista, because cell wall is what the questions are going to come. So here they are telling that cell wall is made up of stiff, okay, stiff cellulose, okay, stiff cellulose plates. They're telling that the cell wall is made up of stiff cellulose plate on the outer surface, and most of them have two flagella. That's what I told you. From the term dino itself, you understand the word dinoflagellates means two flagellas. Okay, one that lies longitudinally and the other transversely in a furrow between the wall plates. Okay, between the wall plates. So one, if I am drawing the dinoflagellates, okay, I'm drawing it like this. One is going longitudinally, one is going transversely, and they are not coming out from some place, they are coming out from a furrow. Let me draw it better to make you understand. Okay, so students, let's draw the dinoflagellates like this. Okay, and let's give it a furrow. Okay, there's a furrow here. Now through this furrow, okay, through this furrow there is one that is coming longitudinally, okay, one flagella that is coming transversely. So they're talking about this furrow. Okay, they're talking about this furrow. So they're telling that the one lies longitudinally and other transversely in a furrow between the wall plates. Very often red dinoflagellates are named as goniolegs. Okay, goniolegs undergo such rapid multiplication that they make the sea appear red or red tides. Toxin released by such large numbers may also kill other marine. So what happens, students? I don't know if all of you have seen or what. Now let's say this is the sea. Okay, this is the sea. I'll try my level best to draw it. Okay, this is, oh no, this doesn't look anything like a sea. Okay, let's just imagine this is the sea. Okay, this is the wave. Got it? This is the wave. Now what is going to happen? This goniolex is a red dinoflagellate. It will multiply rapidly. Rapid multiplication will happen, students. That rapid multiplication, excuse me, okay, that rapid multiplication will lead to making the sea look like red sea. Okay, sea look like red sea. Now what is going to happen? No, this goniolex, yeah, maybe it looks beautiful, but the problem is this goniolex, as it is multiplying rapidly, it is going to release a toxin, and that toxin is going to be harmful for other organisms in the sea. Got it? It is going to be harmful for other organisms in the sea. So that is the main problem. Got it? Dinoflagellates, they appear mostly marine and they are photosynthetic. They appear in YGBBR, that is yellow, brown, blue, green, and red. And depending on the main pigment they have. That is not most important thing. They have a cell wall which is very, very stiff. Okay, they have a stiff cell wall, and they have two flagellas that is arising from a furrow. One flagella is longitudinal, another furrow is another flagella is transverse, and they very often red dinoflagellates called goniolegs rapidly do multiplication and give the appearance of a red sea like appearance, and when they do that, they release a toxin that actually kills other aquatic organisms. Easy. Protista is actually very easy if you understand. Okay, Protista is very easy if you understand. So try to understand as much as possible. Got it? Yes, try to understand as much as possible. Now we will move on to the next one. Next one is euglenoids. Okay, euglena. Euglena, I think everyone knows very well, no? Euglena, ma'am, we have heard 100 times. Okay, so euglena, students, what is going to happen? Majority of them are freshwater organisms and found in stagnant water. Instead of a cell wall, very, very important. This is the thing. Okay, instead of a cell wall, that means they do not have cell wall. They had already told, remember Protista? Okay, okay, imagine, see, who is this? Mulage. Mulage. Am I pronouncing your name right? Yes. Am I pronouncing your name right? Yeah. So students, he is asking what is the difference between poison and toxin. To be frank, toxin is a type of poison itself. Okay, toxin is a type of poison itself, but all the poisons that is there, no, everywhere, it need not be that toxic. Okay, need not be that toxic type of thing. But poison ki one category only is toxic. Okay, toxin. Sorry, not toxic, toxin. Okay, but not necessary that all the poisons we have is very, very has a high range of harming something. Is this clear, Shiv? So here students, instead of a cell wall, okay, instead of a cell wall, what do they have? They have a protein. Okay, they have a protein rich pellicle. Okay, they have a protein rich pellicle. So I told you, as I am again repeating, Protista mainly you have to learn about the cell wall. Okay, so first we studied about cryophytes. You understood that the cell wall is overlapping shells which are soap box shaped, and those cells are made up of silica which are not indestructible. As they keep settling in the seabed, they form diatomaceous earth. Now when you came to dinoflagellates, you got to know that the cell wall is made up of stiff cellulosic plate. Now when you reach euglenoids, you got to know that instead of a cell wall, they have a protein rich layer called as the pellicle. Okay, protein rich layer called as the pellicle, which makes their body flexible. They have two flagella, same thing here. They also have two flagellas. Okay, they also have two flagellas. One is short, one is long. Like we tell, no, every short friend needs a long friend. I mean tall friend. Okay, every tall friend needs a short friend. So same thing here also. They have two flagellas, a short and long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, students, actual onion, all of you know that movie, no, where Vikram will keep changing characters, or like how doppelganger, like how do we tell, dual personality, not doppelganger, dual personality. Same way Euglena has dual personality. Why? I'll tell you. In the presence of sunlight, okay, when sunlight is there, he's like, okay, fine, I'll do autotrophism, that is, I'll make my own food. But the moment sunlight is absent, okay, no sunlight, sunlight is absent, what he will do? He'll be like, I will start feeding. He'll become heterotrophic. He will start feeding smaller organisms. Students, imagine the friends of Euglena. They'll be like, okay, sunlight is there, he's making his own friend. Okay, small organisms will be there. Suddenly, sunlight goes, Euglena starts consuming these people. So, Euglena is proper that, dual personality person, that when in sunlight, they behave as autotrophs, and when they do not have sunlight, they behave as heterotrophs, and they start feeding smaller organisms. Okay, so remember it as a fun thing. Okay, fun thing. Got it? So Euglena will do this. And interestingly, the pigments of Euglena, this is a very important question. Okay, this question has come. Why did we consider Euglena under the plant category? It is mainly because Euglena has the same pigment as higher plants, that is chlorophyll A is there in Euglena as well as in higher plants. Okay, Euglena and higher plants have the same pigment. Got it, all of you? Got it, all of you? Yes. So entirely one green structure I have drawn here. Now slime mold. Okay, slime mold, I told you it acts like a fungi. Right, I told you it acts like a fungi. So slime mold are saprophytic protist, and the body moves along the decaying twigs and lives engulfing organic matter. Under suitable condition, they form very, very, very important line. students, when the condition gets suitable, they will form a structure called as the plasmodium. What they will do? They'll form a structure called as plasmodium. Okay, the structure is called plasmodium. Now, why are they doing this? Because when the condition gets unfavorable, they will form spores and keep, and when the condition gets favorable, they will scatter the spores. Same thing fungi does. So here, during the unfavorable condition, the plasmodium will differentiate. See, the plasmodium will differentiate and form fruiting bodies bearing spores at their tip. So the main job is that when the condition gets unfavorable, it will differentiate. This will differentiate. Okay, this will differentiate and they will form fruiting bodies. They will form fruiting bodies. Now this fruiting bodies will do what? They will bear the spores. When the spores are there, so obviously this fruiting bodies will have like this, no, fruiting bodies. Inside the spores will be there. They will scatter open and they will start going and spreading everywhere. So this is an exact characteristic of fungi. That is why we put slime mold under fungi. Okay, the spores possess true walls. They have true walls, students. The spores, no, have true walls, and they are extremely resistant and survive for many years even at adverse condition. This is the main reason why we call it, no, spores, because they are able to withstand or survive at extreme conditions. Okay, extreme conditions they'll be able to survive. So the spores are dispersed by air currents. So very simple, slime molds are saprophytic. They'll be feeding on dead and decayed organisms. Now under suitable condition, they will form a aggregation. Aggregation means what? Group. Okay, group. A group of structures called as plasmodium. That plasmodium will differentiate when the condition gets unfavorable. Okay, remember, favorable condition, I will keep money for myself, or I will collect rice and keep for myself. When the condition gets unfavorable, I will utilize this rice, I will utilize this money to feed myself. All of you know that ant story, how the ants collect grasshopper or ant, one among them, they'll collect the food grains and they'll keep storing it, and the other person will ask, why are you storing it? It's not even rainy season yet. The ant will be like, no, I am preparing myself for the rainy season. And then this ant will keep collecting in the month of summer. Grasshopper will wait for the rainy season, and in the rainy season the grasshopper will have no food. From where to where I went. So the ants, no, like the ants only, these people also, during the condition, when the condition is favorable, they'll make an aggregation called the plasmodium. When the condition gets unfavorable, they will break this plasmodium, or the plasmodium will differentiate by itself. It will have fruiting bodies, and inside this fruiting bodies, spores will be there. This spores will be scattered with the help of air currents. And the spores, students, because it has to scatter, it has to deviate, and so, okay, this will deviate, deviate, and it will go fall somewhere, because of that they need the cell wall, otherwise this will gone. Okay, imagine no protection is there, obviously it will go and fall somewhere and it will get damaged. So they'll have proper cell wall. Roshni, did the ants actually save the food or the grasshopper saved the food? I'm not able to remember who was the good person in the story. Okay, who suffered in the last, I'm not able to understand. So during unfavorable condition this happened, during suitable condition they will form plasmodium. Okay, yes, done, done. Sometimes these stories are very beautiful, no, like they give you a lot of like they make you understand in the best way possible. So I always link my class to so many stories, so many, so many things, so the students like, enjoy it. So protozoan, students, all protozoans are heterotrophs. Okay, all protozoans are heterotrophs, because of which we put them into animal category. Okay, we put them into animal category, and they live as predators or as parasites. They are ants only, ma'am. Okay, so ants were the ones who actually saved the food, and the grasshopper was left with no food. Okay, they are believed to be primitive relatives of animals. Why they are primitive relatives? Because you notice that all these people, amoeboids, ciliated, all of them do some kind of locomotion. At the same time, they are heterotrophs. Now this two category is the biggest category of why we can call it an animal, no, that they are able to do locomotion. At the same time, they are also not able to make their own food. They depend on other organism for food. Yes. Okay, so the first category is called as amoeboid protozoans. Okay, these organisms live in freshwater, seawater, or moist soil. Okay, freshwater, seawater, or moist soil. They move and capture. Okay, so what do? Amoeboid means what, students? Amoeba. Okay, amoeba, they have pseudopodia. They have pseudopodia, or I can call it in other words, false feet, ma'am. Yes or no? I will call it false feet. That is, they will use this false feed to fetch something. So amoeboid protozoans, these organisms live in freshwater, seawater or moist soil. They move and capture their prey. Okay, they move and capture their food, whatever they want to feed on with the help of pseudopodia. So look here, by their prey, by putting out pseudopodia, that is the false feed as in amoeba. Marine forms have silica shells, very important, okay. Marine, excuse me, okay, marine forms, students, marine forms have silica. Marine forms have silica shells on their surface. Some of them, such as Entamoeba histolytica. Now you will learn this Entamoeba histolytica tomorrow in my human health and diseases chapter. Entamoeba histolytica is the one, okay, that causes amoebiasis. You go and eat no outside all this pani puri, that this golgappa and all that, that will, he would have made it with contaminated water. Not everyone, but some people do it with contaminated water. Now when you consume that, what happens, your body will have the any kind of germs or any kind of bacteria or pathogenic bacteria. Now if you or any other person for that matter does not leave their or leave their excreta in a proper place, like proper sanitary, everything is not connected, or this excreta is going and mixing with the soils. Now in the soils the vegetables will be grown, in the vegetables contamination will be there, and that vegetable comes to the market and we consume it. Now I don't know how many of you have noticed, normally also if you notice that even though every day you're eating food at home and you're not eating any junk food also, sometimes you'll notice that you are affected with diarrhea, or you have constipation, some or the other problems. Yes or no? That could be because of this Entamoeba histolytica. That is the farmers, right? Like imagine, students, in some village areas where there is no proper sanitization, there's no proper sewage system, many people actually do their excreta in open lands. Now I'm not telling it is necessary that it will they will get into a farmer's place only and do it, because proper fence will be done. But other ways, generally if the farmer is doing a very small cultivation or something, cabbage or something, if there are people, if maybe their own family members are doing the excreta, if that person is affected by contaminated food, then there is chance that when these vegetables come to market, we are also affected. That could be a reason why when we eat normally at home also, normal vegetables also, we might fall sick. Okay, that is because of contamination, and that is caused by Entamoeba histolytica. Now flagellated protozoans, from the name itself you know flagella they have. Okay, flagella. So the members of this group are neither free living or parasitic. That is, they're not completely free living or they are parasitic. They have flagella. The parasitic form causes diseases such as sleeping sickness, which is called as trypanosoma. This you have to remember. Okay, this you have to remember. Flagellated protozoans means they have flagella. Amoeboid protozoans means they have pseudopodia. Okay, don't write it as amoeba. Like amoeba, they have pseudopodia. Next is ciliated protozoans. Okay, ciliated protozoans, from the term name itself you understand cilia is that. Okay, ciliated protozoans, they are aquatic, actively moving organisms because of the presence of thousands of cilia. Yes or no? Many cilias are there. Okay, they have a cavity called the gullet that opens to the outside of the cell surface. That is, students, if all of you have seen, the example for this is paramecium. Okay, I'll try drawing paramecium. Okay, this is paramecium. It will look like the sole of your shoe. Now here, if in your NCERT, you can see that there is a small bending like this towards the inside. That is called the gullet or the oral cavity. Okay, that is where the cavity. Okay, that cavity is called as the gullet. Okay, gullet. So what are they telling? They have a cavity called the gullet that opens outside to the cell surface. So anything is there, it will come out with the help of this gullet cavity on the cell surface. The coordinated movements of rows of cilia causes the water laden with food to be stirred into the gullet. So entry also, exit also is from the same place. That is, food is entering through the gullet. Now I'm not able to write it here, so I'll draw it again above. Okay, food is entering also. Students, like this, let's draw it here. Not so sharp also, but yeah. Okay, it will look like the sole of your shoe. So this is how your paramecium looks like. Imagine. Okay, this is the cavity. This portion is the cavity. I have drawn it little inside. Okay. Yes, this is the cavity. Now the food will move. Okay. Everywhere cilia is there. Okay, everywhere cilia is there. All this place cilia is there. Now what is going to happen? The cilia will bring the food. Okay, this is the food. The cilia will bring the food and it will allow the entry through the gullet. Okay, gullet is the small cavity. Now even exit, that is after utilization of the food, no, whatever is the excreta waste materials also will come out through the gullet itself. So that is what they're trying to say. Okay, example is paramecium. Paramecium. Okay, paramecium. Now sporozoans. What is sporozoans? This include diverse organisms that have an infectious spore like stage. Students, here nothing about locomotion is spoken. Understand that spores are the ones who are moving away. Okay, one place spores only will be moving away. All of you mark your attendance, smash that like button. Why are you doing conjusi? Whenever you're watching a lecture, always make it a point to hit that like button. Okay, this includes diverse organisms that have an infectious spore like stage in their life cycle. The most notorious is plasmodium. Okay, the one that causes malaria. Okay, plasmodium vivax, full name is plasmodium vivax. Plasmodium vivax. Student, this is the one that causes malaria, and this is one of the most dangerous one. And plasmodium falciparum is the most malignant, like very scary one. Okay, very scary one. This causes malaria, a disease which has a staggering effect on human population. So sporozoans means what? They have one of the stage where there is spores present. Okay, understood. So here they're talking about the spore stages, sporozoids. If all of you remember malaria, you know that in the mosquito there is sporozoids. Yes or no? That will be stored in the salivary, that will be kept in the salivary gland, and it will inject a person, and from that person the sporozoids will go. It will multiply in the liver cells. Liver cells will burst open. From there it will go to the RBC cells will burst open, and that will release a enzyme called as hemozoin. Okay, what will it release? Hemozoin. This hemozoin will cause fever and chills. This hemozoin will cause fever and chills. Yes or no? Understood. Now this fever and chills will be caused by the hemozoin. Now this what happens now, students? This gamete formation will happen in the body of the host, that is humans. But fusion will not happen. Understood? Fusion will not happen. That is fertilization will not happen in our body. So this mosquito will come, maybe another mosquito, maybe the same mosquito, who knows. That mosquito will come, it will suck our blood. In the blood the sporozoites will be there. Now these gametes will fertilize in the gut of the mosquito, and they will be stored here. They will be kept in the salivary gland, and when the time comes they'll go and bite another person. So this is a cycle of malaria, and it is caused by plasmodium. So here what are they telling? This includes diverse organism that has an infectious spore like stage. So the spore like stage is this sporozoid stage, which you will see in the mosquito, and that will be passed on to us. Okay, passed on to us. Done. Yes or no? Okay. So here I am going to write some things about Protista. Protista. Ready? Chalo. So this Protista, 5 categories there. 1, 2, 3, 4, 5. No, this is not possible. Okay, let's do one thing, students. Let's take this Protista to the center. Okay, let's take this Protista to the center. Easy chapter, okay, but I don't know why students find it very difficult. I think I'll find it difficult to write this. Let's try. Cryophytes. Chrysophytes also you can tell. Chrysophytes. Okay. Dinoflagellates. Dinoflagellates. Okay, from there you come here. You have euglenoids. Okay, from euglenoids you come further. You have slime mold. From slime mold you come further. You have sporozoans. Sorry, protozoans. Yes, clear? Okay, awesome. So, 1 second, students. Voice is fine only, no Roshni? Yes, here what is going to happen? That is chrysophyte. I'm only going to write about the cell wall. Okay, that is the most important thing that you have to know. See, in chrysophytes, you have to speak about diatoms. Okay, in diatoms, you have to speak about the overlapping cell wall. Okay, overlapping cell wall, and you have to remember that this overlapping cell wall is made up of silica, and this silica gets settled for a longer time and form the diatomaceous earth. I'm writing only the main words. Okay, diatomaceous earth. Good. This diatomaceous earth will be used for filtering, polishing, etc., etc., etc. Okay, polishing, and they are the chief producers in the ocean. Okay, now next one is dinoflagellates. Dinoflagellates are especially main thing again, cell wall is most important thing. Cell wall is made up of stiff cellulose. Stiff cellulose. Okay, and they have two flagella. Okay, two flagella. One longitudinal, one transverse. Okay, one longitudinal, one transverse, and these two flagellas also comes from the furrow. Okay, furrow, and then they have goniolex. Okay, goniolex. Students, color you remember. Okay, YBGBR. I wrote it a little ulta. Yes, ma'am, audible. Okay, goniolex is the red dinoflagellate. Okay, it will form the red tide. Okay, red tide appearance, and also they will release a toxin that actually kills smaller organisms. Okay, done, done, done. Then we have euglenoids. Euglenoids does not have cell wall, no cell wall. They had already mentioned, remember, Protista, not everyone has the cell wall. Okay, so instead of cell wall, they have a protein rich layer. Okay, protein rich layer called as the pellicle. What is it called? Pellicle. And then we notice that in the presence of sunlight, okay, when sunlight is there, they are autotrophs. When no sunlight, okay, sunlight is absent, they are heterotrophs. They are heterotrophs. Okay, then what else, students? These people also have flagella. One long flagella, and short flagella. Okay, short flagella. Yes, got it. This is what overview. Then slime mold, we'll write it here. Okay, slime mold. I'm giving you revision also, so you should be in a good position to remember all this tomorrow as well. Okay, slime mold, students, main thing is when the condition is suitable. Okay, when the condition is suitable, they form an aggregation called as plasmodium. Okay, aggregation called as plasmodium. And when the condition gets unfavorable, okay, when the condition gets unfavorable, they form the plasmodium differentiates. Okay, plasmodium differentiates and forms spores. From where did the spores come? It came from the fruiting body. Okay, it came from the fruiting body. Done, done, done. Okay, it came from the fruiting body, and then they will get scattered. They are heterotrophic. They behave just like your fungi. Okay, now protozoan, students, protozoans, you have categories like amoeboid. Okay, amoeboid protozoan. You have category called as flagellated. Yes or no? Flagellated. Then you have category called as ciliated. Then you have category called as sporozoans. So amoeboid ones have pseudopodia. Okay, Example, where to write example, students? Okay, example is Entamoeba histolytica. Okay, example is Entamoeba histolytica. Then flagellated, you have flagella. Okay, you have flagella. Example is trypanosoma. Okay, trypanosoma. One second. Okay, yes, trypanosoma. Okay, flagella, trypanosoma. In cilia, you have cilia. Sorry, in ciliated, you have cilia. Example, you have to remember paramecium. Okay, paramecium. And here you should also remember the word gullet. Okay, that is a cavity. That is a cavity. Okay, then sporozoans, they have a spore like stage. Okay, spore like stage. And example is going to be plasmodium vivax, that causes malaria. It causes malaria. Done. Yes or no? So we did Protista, we did Monera. Okay. Now students, this is more than enough for you. You do not have to refer hundreds of books. This information is more than enough. Okay, more than enough. This is solid information. Okay, that is more than enough. Okay, plasmodium differentiated, yes, to form spores. Okay, plasmodium vivax, done. Students, this plasmodium and this plasmodium, don't get confused. This is an organism called as plasmodium vivax, and this is an aggregation. Okay, this is an aggregation that is formed like this, and inside that they have spores. Okay, lot of forex, I mean lot of differences there. Okay, lot of differences there. Yes, so we have done. So you can just see we have wrote all of it as well. See, we have understood every line of NCERT. So one second, I'll just stop under Monera. See, here I've written everything about Monera. There I've written everything about Protista, and also every line of NCERT as well. More than this, you do not need anything. So just focus on this and try to learn as much as possible. So this way what happens is you will be able to understand NCERT line by line, and this will be an everyday series. You will get everyday same content like this, like one chapter we'll be trying to finish. Next class will be fungi. So by fungi we'll be done with this chapter, and then we'll move to the next chapter as well. Okay, so I hope the session was helpful. Definitely let me know in the comments, and also smash the like button, and then give me the motivation to come here every day and teach you guys. Okay, so I'm super happy to teach you, all of you, and give this free content on YouTube, because what happens is on YouTube, you know that there's so much content, but it is very necessary that the content that we put out is helpful for the students. It's in a simpler language, and students are able to connect and understand. So I think that is the most important thing, and Vedantu has been doing that. So yes, that's it for today. So see you all in the next class. Thank you so much for joining, and Gopika ma'am signing off. That is so see you all in the next class with the next topic, that is fungi. Fungi we have little more in depth, not like Monera and Protista. Little more in depth we have classes, we have about each of them we have to learn. So